Because in this paper, if you read it like you were supposed to, you're going to see that the stuff we did this past summer, so freshmen, this isn't about you, but the stuff we did this past summer actually should help you understand what they did in this paper, because it's a big part.
All right, so I did paper on generation of scalable cancer models by combining AAV intron trap, CRISPR-Cas9, and inducible Cray recombinase. And some background you need to know for this is what CRISPR-Cas9 is and what it does. CRISPR-Cas9 is a gene editing tool that is typically used to knock out genes. And as you can see in the diagram, it has basically two components, the CRISPR guide RNA and the Cas9 protein. And those two form the Cas9 complex. And now the Cas9 will locate a genome or a specific gene sequence in the genome using the guide RNA, and it will find a certain section on that genome called a PAM. And once that PAM is found, the guide RNA will bind itself to the DNA target sequence, like you can see in the diagram. And once it is paired, the Cas9 protein has two nucleases which snip each end of the DNA, or each strand, and it causes a double strand break in the DNA. And after it does that, its job is basically done. And when the cell goes to basically fix that break in the DNA, oftentimes it results in mutations, and it can't fix that segment back to how it was. And so it oftentimes knocks out that specific gene, and it can't be used for anything again. And that's the base of CRISPR, but a lot of people have tried or have been adding other proteins and enzymes onto the Cas9 protein, and they've also been shutting off the nucleases on that protein, and so they could do single base pair edits. So they could use the Cas9 protein and the guide RNA to find a certain sequence, and they could add an enzyme or a protein that can knock off and substitute a different base pair for another, which can cause a non-synonymous mutation, or basically a mutation where if one base pair is changed, that 3 base codon is then changed, and it will have a different amino acid being produced from that DNA, from that translation, and it will form a completely different protein and cause possibly physiological changes.
Okay, so I'm going to interrupt, if you don't mind.
Okay.
Do you mind? Is that all right?
That's fine.
That is really well said. Okay, so those of you who've had honors biology, and especially if you've had AP biology, should better understand what he just said, because it was really well put, right? In other words, sequence equals structure equals function, and you're actually using a tool to change the sequence and making a non-synonymous change in the codon, so it becomes another amino acid, so the protein has a different shape and therefore has a different function. Right? That's pretty much what he just said. Are you all on the same page now? Right? Okay. Because, go ahead, take it away, because I think you're going to get to the implication right away, right?
Okay. Yeah, basically. Well, just one more thing about CRISPR is if you do not add those extra proteins on and you simply cut the DNA, sometimes it's pretty rare, but sometimes the response of the cell when it fixes that double strand break, it can result in a mutation, which could cause a toxic gene, which could produce protein that causes the cell to have it may not reproduce properly. It may not. It just won't work the same. And so that's one thing they identified in this paper was CRISPR couldn't be used to edit a certain gene in cancer cells. And so they had to develop a new technique to edit that gene and detoxify it in a way. And so the main thing they needed was a vector to get the CRISPR into those cells, and they needed that vector, and they needed to combine a bunch of things to make this system work and be able to detoxify that gene. And so we go on to figure 1, and figure 1 is basically the creation of that the donor. It's called a recombinant. Let me just get this right. Recombinant AAV, or recombinant adeno-associated virus. And so it's just a certain strand of synthetic DNA that they created to be inserted into the cell and basically edit the genome where they had the toxic gene, if that makes sense. And then the process they used to get this large sequence of DNA in is quite confusing. They basically had to use CRISPR to cut the DNA, and then they took advantage of the homology. They took advantage of the homology-directed repair, which is the repair of that double-stranded break, and they were able to force the cell into incorporating that large portion of that large chunk of DNA in between the two strands, basically. And so they went and tested it. And figure 2, don't worry about part A. It's a very confusing figure. It doesn't make much sense. But part B, Dr. Tra, I don't know if you're there. Okay, he's not there.
I am. I'm listening to you. I just had to go off camera for a moment. So, what was your question?
Oh no, I just don't remember exactly what B and C were for.
Oh, so B and C were showing. Let's see. B and C were showing what the effects would be, and what's cool about of doing what's in A. So the breakdown of what the single site target would be in the DNA, and then to make sure that it had worked, they're running PCR and looking at the gels in B and C. So that should look familiar to you guys from the summer, because that's what we did, right? So, Sean, can you explain what the different columns are?
Yeah, so on the far left, they have what's called a ladder, and they didn't label this, or they didn't explain it, that's not the greatest thing to do in a paper. It makes it very confusing, but it's a ladder, and it basically shows where different, how do I explain it? Like different levels on the gel where you could find
size. It's size.
Oh yeah, it's like different levels for different sizes of the DNA strands. And so in the middle, or just to the right of that ladder, they have a control sequence, which is an unedited piece, an unedited portion from the genome. And then to the right of that, they have the HDR, which is the homology-directed response piece. I don't know how to explain that. And it just shows how there is another portion of DNA. Is that
right? So there's the edited, so in the heterozygote, like we said, we did this exact thing over the summer. If you were a het, you'd have a band at one area because it was a certain size, and then you'd have another band that was smaller that had gone down farther on the gel because it was smaller, it was faster. So it'd travel more distance in the same amount of time, faster. So you'd have two bands. So what you're looking at is the unedited version, and that's actually, right, unrecombined is what it's called, and that's 1.5. And then the cut version, which is recombined. And if you look below it, you see that there's one that doesn't have Cre, and then one that does have Cre. And what Cre does, Cre recombinase is an enzyme that when it's activated will go and cut the gene at certain sites, which is why you see multiple bands there. So there's a couple things I wanted to add to your background. Molecular cloning, molecular biology has been around since the 70s and has usually involved altering groups of bases in DNA in a single-stranded way, right? Because now this is going to be a little over the freshman's head, but the rest of you should get this. In DNA, it's a double helix. It's one strand and another strand running parallel and twisting around each other, right? And so the way this has always been done, I can actually draw this, is how we did it. We never did it because of COVID. Thanks, COVID. People who are in AP, we're going to do this lab pretty soon, but it's this sticky ends idea. So I don't know if you can see that. Yeah, you can see that. So let's say you have GAATC, and it continues ATGCCA, and you could go before it like TCA. I'm just making that up, right? So that's a sequence on one of the strands. The opposite strand will be its complement. So T, G, A, C, T, T, A, G, T, C, G, G, T, right? So pretty easy based on base pairing. Come on, focus. Okay. And what we can do ever since the 70s is be able to get restriction enzymes, so they're enzymes, they do stuff, that make a cut at exactly this, it recognizes GAATC, and it'll cut it in such a way that I now have, if I were to draw it like this, it would kind of look like this, like kind of a sticky end. It would have a series of stuff, and then the other one would be its complement, like that. So they come together kind of like Legos. So you're able to like edit a single thing or a single thing, but that means it's kind of inefficient. Doing a double strand where you're actually taking some DNA, right? DNA has all of these bases running in parallel, and you actually say, I want to cut right here and here, and double strand break. That is a new thing in CRISPR, that or a new way that you can use CRISPR that makes it really unique, because as Sean just said, your cell will actually try to repair the damage as best it can using the template it has. But if it's a double strand break, it doesn't have a template because there's no complement to it. So you actually get a change, which is a failure to fix it, which is a good thing, right? You get a change that can't be fixed. Johan, what'd you want?
I was just clarifying something, but I think you explained it. You were just saying that the DNA is like broken up right through the strand. It's not like they like separate the helix and then do the editing, like you were saying. They just cut through both strands and then.
Yeah, it's less intense. It's easier to pull off than if you're talking about DNA replication or transcription, where you have to unwind it and you don't have to do that. This thing just goes, you know what, and just cuts it. So that makes it way more efficient because it doesn't take any extra effort to unwind anything. You just make a cut. You find it, make a cut. The other thing I wanted to say was AAV, the adenovirus, is also another old way to get a piece of DNA into another living thing. So whenever you take, we learn this in honors, I believe. When you take a piece of DNA from one species and you put it into another species, what is that called? I know you guys know what it is because you try to avoid the food that has this.
GMO?
Correct. Genetically modified organism. Okay, it's also called a transgenic. So genic genes that are being transferred into another species. So transgenic. The process is actually called cloning. So if you're wondering, like, wait, I thought cloning was taking one thing and making a copy of it. Well, yeah, that could be the result, but it had to be done using the process we call molecular cloning. Molecular biology as a field is actually based on this one strategy. So to take DNA from one living organism and be able to stick it into another organism is called cloning. It's done by molecular biology, and the way that we do it is by putting DNA into what we call a vector. So you can think of a vector as like a little bus or a ship or something to get DNA from one organism to another. We have found over the years that some of the most efficient vectors are actually viruses. They're really, really good at packaging DNA so that you can put them in cells. Adenovirus, AAV, is actually probably the most commonly used one. It's really efficient. It's not super good at getting in neurons, but it is pretty good at everything else. So if you're wondering, like, well, then how do you get it into neurons if you want to? You got to use a different virus, something like HIV. HIV/AIDS is a lentivirus that is really good at infecting neurons. So we actually use that instead. I think that's all I wanted to say. Any questions? Is that interesting? Okay. All right, keep going, Sean.
All right. And just to bridge off of what Dr. Char said about the vectors, a lot of the times they'll take out the DNA or RNA inside the vector, which is used to reproduce that virus, and they'll just put what they need to get into the cell. So in this case, they just had it was some really funky stuff they used to make CRISPR, because they couldn't actually fit CRISPR inside the vector, but they just put CRISPR and the other gene they were inserting, the transgene, inside that vector, and they didn't have anything else in it, because all they had to do was just get the stuff to the cell, and that was it. And so in figure 2, again, we see how by cutting the DNA and inserting that transgene, they were able to they were able to mutate one base pair, which was able to turn on the other half of the heterozygous gene. So the gene they were testing is heterozygous, and typically one of those genes is on and the other one's off, and they were able to turn the one that is off on, and so that's why we see the 2 strands in figure B on figure 2. And then when you look at D, we see how that base was edited. It's the graph on D is just very unclear. Dr. and I saw that.
Yeah, Sean and I were staring at this because I know how to read this because I used to run sequencing all the time, but I don't think it's fair to not label the colors for you. Like, the colors actually mean something. And it actually is interesting.
It has the pieces in.
Oh, so yeah, that's true. I guess you could just use that.
And that's later in the paper. That wouldn't.
Yeah. Yeah, it should come first, though. Listen, if you're writing a paper, AP Biology people, if you're writing a manuscript, you wanna explain what the person's looking at in the figure the first time it shows up, not like 3 figures later. So I don't know. I know how to read D, but most people won't. Johan.
So this may be a dumb question, but
Stop.
so in figure E, or is that no that in figure C?
Yeah.
So there are like 3 DNA bands, right? Ladders, I mean. My bad. And I don't know what the CRE, like negative CRE and positive CRE are. Like which ones are those?
You want to take that, Sean, or you want me to do
it? You can do that one.
So Cre is a type of enzyme. Oh, that was the other thing I wanted to tell you guys about. Was the origin of CRISPR, like where it comes from. Do you want to say anything about that, Sean? Like where did Jennifer Doudna actually get CRISPR from in the first place?
It was from bacterial immune systems, right?
Yes.
Yeah.
It's like the most primitive yet highly effective type of immune system you can possibly have, because single cells use it to attack viruses. It recognizes a viral sequence and then slices it up so that it can't be used. Okay? Now, Cre is another one of those similar enzymes that is designed to cut at only a part of the genome that's called LOX. It's called a LOX-P site. And so what you do is you design a piece of DNA that has a LOX on one side and a LOX on the other. The process is called floxing, to flox the gene. And this is pretty cool because you can do it in a way that's temporary, where you can actually turn a gene on or off with like an antibiotic or something. So you can control it and see what the difference is. So like AP Bio people who we just did statistics with. Remember how we talked about matched samples versus independent samples? And how matched samples are better? They're more powerful statistically. If you can actually have an animal that expresses a gene and you measure its behavior, and then give them a drug that turns that gene off, you can then measure that same animal again. Do you see how powerful that is? Right? And then you, as a control, you could then turn the gene back on and measure them again and say, yeah, it restored the behavior or the physiological process from before, showing us that this gene does this, which may seem obvious to you guys who are living in the 21st century, but in the 20th century, we weren't sure about stuff like that. We didn't know that a gene made a protein which caused a behavior. So Cre is a really precise way to cut a part of the gene out when you want it to be cut out. And so if you look at let's see, what is that? C, the negative Cre means that it hasn't been used. That's the negative control. Versus the positive Cre means that you've actually activated the Cre and it's gone and cut the gene where it's supposed to, creating different alleles.
Now, I'm assuming that. My bad.
I said it's like you're creating evolution, right? Something that would have happened naturally over millions of years, you can do now in a lab by just turning on Cre. Go ahead, Johan.
I'm assuming that first DNA ladder is CRISPR. Right. What do you say?
Are you talking about the
no, the bottom one,
right of the left? Or
I mean, yeah, in that row.
Okay, so you the first row is
Sorry, we had a baby fall. If he wasn't related to me, I would have laughed. But all right, so say that again. What were you saying, Johan? Sorry, yeah, distracted.
I just was asking if the first DNA ladder was CRISPR,
No,
and
so
then
the ladder in the first column is
yeah,
the same for B and C. Can you turn that down
right.
or off? Yeah, thanks. Just the audio. Yeah, just the audio. It's the same, and all it's doing is it has bands of a certain size. Smaller size things travel farther through the gel, just like we did in the summer. We did this experiment practically.
With the electrophoresis thing.
Right. So that's an electrophoretic gel. That's the same. It's agarose. It's the same exact thing you did, except that the reason it's black is, well, it's pretty much the same thing. They used a dye just like we did. We put a dye on it. It made the DNA came up as blue, you know. But the important thing here is that the result of C is what happens in D, which Sean can't really tell you what that is until a later figure because they did such a bad job labeling it. Right? So, Sean, go ahead and jump to the next one. Yeah, because I think this one, right, this one's probably the most important figure.
Yeah.
Yeah.
Yeah, so in figure 3, they show the...
Hit present, Sean. Yeah, that's better.
Okay, in figure 3, they show the gene therapy just using the adeno-associated virus. And that's in A, B, and C. So A is the PCR result of those cells that were affected by the adeno-associated virus. And
right, remember, hold on, Sean. Remember over the summer, because we had limited time, we actually ran like a row. We were going to run one row and then directly under it on the same gel, we ran another one. I remember. Is Sarah McTighe here? Sarah, you here? I remember she was helping do this, like set it up. So we had multiple rows running on the same gel at the same time. So that's all you're looking at here. Okay, they're running over the same time in the same gel. Ladders over on the left. Go ahead, Sean.
Okay, so there's 3 rows, and they tested 25 cells total, and only 4 of which had the mutation which they were trying to test or create, and that was like only what 16% of all those cells were able to show that mutation. And you can see in figure in B, they show how there is no edit in the genome, or no edit in the bases of those cells. But then we go to D, where they tested it with the AAV and CRISPR combined. You can see that 23 out of the 25 cells infected with that system had the mutation, which resulted in about a 92% efficiency. And if you look in figure D, or no, figure E, you can see this figure is really screwed up. But you could see in the 7th, is it the 7th? In the 7th peak, it goes from an A, as it is in the in B, and it goes black. It goes to a G. So they were able to
both. It goes to both. You can see it's somewhat green. It's a 50-50 mix. Yeah.
They were able to
change, right? You can see there's 2 bands. If it was just G, you would have only gotten the lower band.
Oh yeah, I forgot.
But it wasn't. It was 50-50. Yeah.
Mm-hmm. Yeah.
Which is still incredible. That's incredible.
Now that figure is really confusing because first off, they got a dotted line in there which has no significance to the figure whatsoever, and they labeled in B they labeled the A over the wrong base, and then in E again they labeled it over the wrong base again. It should have been over the 7th one.
Just that mistake, those two mistakes, I probably would have rejected the paper. I would have sent it back to the authors and I would have said, listen, if you're this sloppy in your figures, then how do I know that you did the rest of the experiments right? Like this is an important lesson for all of you, but especially the new people, the freshmen, right? How precise you are or how careful you are when you do your work matters. You do not want to leave the bad impression that you have no idea what you're doing. This, like Sean said, this is screwed up. The A label's in the wrong spot, and then he hasn't told you about E. Go ahead and tell him about E.
Oh, E?
Yeah.
Well, yeah, it's screwed up.
That's not. If they didn't even label it in the same wrong spot. They put it in a different wrong spot. That's not where it is. That's stupid. So, and at this point, right? At this point in Figure 3, we still don't know, unless you're used to reading Sanger sequencing, you don't know what colors what really, right? Because that's Figure 4. So for those of you who are interested, if I look at this, someone who's done this a lot, blue means C, so cytosine. Green is A because it's labeled, right? Black is G, so it's guanine, and T is thymine. So it goes CAG, CA, G. The original is AAA, GTC, GTTC. But if you go back up, you'll see that that at that 7th position in E, it's a 50-50 mix of G and A, which is cool. So that's the very nature of what a heterozygote is. Yeah.
Mm-hmm. So
one base. That's it. One base did that major change.
Yeah. So from here, I mean, they were able to show that there was a change in the DNA, but that's not really important in this paper. What's really important when you talk about a change in DNA is the change in the protein and the physiological effects of the change in the proteins. And so in Figure 4, they show in B how the SF3B1 protein has been is produced way more than it is without this without the introduction of the system. And they screwed up this figure again because they put the post-induction, which is after they've placed the system in the cells, before the pre-induction, and it makes it really confusing if you go through it fast. So if you look at it, well, first off, the control in this is GAPDH, which is a protein that's found in basically all cells. And so they used that as a control to show how it has the same levels pre and post induction. And then they used the experimental SF3B1 protein to show how pre-induction there wasn't much protein produced, and how post-induction the production of that protein was way higher. And this, if you think this or C is a PCR test, it's not. It's a different test for proteins. I don't remember the exact name of it. What was?
Western blot.
Oh yeah, Western blot.
Yeah, so these gels, while they look like the electrophoresis that we did over the summer, instead of being on DNA like we did this summer, this is actually with protein. And protein, it's still the same idea, right? Proteins have a charge. They are run through a gel, just like the DNA, except that now you're looking at the amount of a certain protein. And yeah, causes come before effects usually if you're actually in reality. So I don't like that they switched them. But the important thing is that this is protein, a measurement of whole protein in a cell rather than just DNA.
Yeah, so that's basically figure 4. Do I have to explain C? I don't.
I think as part of your implications, it's important, but you should go to your implications first and then come back to it.
So from this, why is this blank? Hold on. Oh, you can see it now. So this paper, it brings us a steps toward genomic surgery and therapeutics, and it brings us closer to a possible cure for cancer, as being able to get this system into cells and change certain genes to express different proteins. That's very important, because if you are able to get into cancer cells and edit certain cancerous genes that are specific to those cancer cells, you could potentially cause apoptosis and terminate all those cancer cells, basically curing the cancer. So,
Can you say that again?
through this technique, they're bringing us a step closer to a cure for cancer, because this technique allows us to get into cancer cells, edit a certain gene to express a certain protein that is, it's a cancerous gene. So if you were to inject this into your body and you did not have cancer, it wouldn't affect you. But if you have cancer and you were to get this system into you and it was able to spread throughout all your cells, it would only affect cancerous cells with those cancerous genes, and it would be able to edit the proteins that are produced by that. And just being able to do that and cause a physiological change, you could potentially cause apoptosis, or basically cell death in those cancer cells, and cure cancer from this.
Yeah, you tell the cell, hey, turn on your kill switch.
Yeah.
And then the cell goes, okay, and commits suicide. In a nice, that's different from, like, when you have a stroke, and you've all had strokes, right? When you have a stroke and all of a sudden parts of your brain aren't getting oxygen and so they explode, that's not what apoptosis is. Apoptosis is like very, very organized seppuku. They commit suicide and they bleb off into different parts, and it's nice and easy to clean up and it doesn't do any further damage. And so, yeah, but are there complications, Sean, with this as it is right now? This is where it goes back into 4.
Oh yeah, I don't remember exactly what C was about in 4.
So go back to present mode and then go to your first slide. So the title of this is Generation of Scalable Cancer Models by Combining AAV Intron Trap. Anybody look up what an intron trap is? Anybody? I actually had to look it up. I actually did not know what it was. I had to look it up beforehand. And it's kind of cool. An intron trap is when you stick in an enhancer, gene enhancer, into an intron to make it more active, to make the gene more heavily expressed. Okay? Like, if you remember, for honors, this would be chapter 11, I think. Yeah, I think it's 11. It's where so a gene has a promoter, and the promoter gets enhanced by another piece of DNA that's called an enhancer. you'd be sticking this enhancer, an extra enhancer in to cause it to produce more of itself. But the thing is that enhancers are really far away from promoters, and the DNA gets folded over onto the promoter. But that means that it can also get folded on to other promoters because it's really far away. So if you go now to figure 4, Sean, what they did was they looked at other genes in the area, other genes that were close to it that might also see an enhanced effect, and saw that it did produce an enhanced effect in protein. So this again is a Western blot, and it's showing that other proteins were also enhanced, not just the one that they targeted. And you could see how that may not be necessarily a good thing. Does that make sense? Like, you don't want to be turning on things haphazardly. You just want it to be very ultra precise, just the gene that I want. I want to turn on apoptosis only in cells that are also doing this other thing, cancer.
Mm-hmm. And these other proteins they aren't caused by cancer, right?
No, they're just I think they happen to just be around the gene of interest, the SF3B1 gene. So because that one was mutated, all these other genes downstream are also getting infected. Yeah. Now I've said for the last 5 years that CRISPR is why we're doing it first, because it's going to end up probably being a method that comes up in multiple journal clubs. CRISPR is the most important discovery of my lifetime, and because the implications for that, the implications of that are that that you can use it to target specific things, and cancer, of course, is obviously one of them. We're not quite there yet, but this is another big step towards being able to do that. Which is why it's interesting. Which is why I approved it. Go ahead and stop presenting, Sean, so I can see other people's faces. Okay. Again, the rule, people, is you keep your camera on. So stop. And I want to see you. I don't want to see your ceiling. All right. So, any questions? The future journal clubs are not going to be like this. It's going to be much more you guys getting involved and handling figures in a Socratic way. We're going to do it like a real journal club. We're going to get used to that. And the seniors, they have their capstone in the 3rd quarter, so that's pretty intense. That's going to be hard. To do this and the capstone will be, actually, it might make doing the capstone may make this easier because you'll be reading a lot of papers. But those of you who are presenting, if you need a paper, you just need to send me the PM ID and I'll send it back to you right away. And then as soon as we decide on what the paper is, it goes on the website and in Schoology, both website and Schoology, so that everyone can access it. Okay, along with any other tools you might find useful. Okay. Any other questions? I hope that wasn't torture. Vanya.
How long does it take for that to happen? Is it I don't know how long it would take. If that question makes sense.
You're interrupting my meeting, Samson. Come here. Come on. Come here. Come on. It'll take him a while to get over here. How long does it take? Sean, did they? That was only in a dish, right?
Yeah, a couple things took
Come on. Come on.
I don't know how to explain this.
Keep going.
Some of the processes took a day or so. They mentioned something taking roughly 10 days to do. I don't remember exactly. Yes. Cloning of the arms. No. So it's like roughly a day.
Okay.
But then again, the culture was only a few cells. So if you were
Yeah.
And it was
on a
a
human,
monolayer of cells in a dish. That's going to be different than a thick tumor made of billions of cells, which is why the vector is so important. Which vector you choose, how infectious it is. As he coughs in my face, I said the vector is so important, how infectious it is, and he goes right in my face. Thank you. Like the new Delta variant of COVID, of the SARS-CoV-2, is way more infectious than the original variant, which was already really infectious, and so that way it can infect lots and lots of cells in a little bit of time. And so you finding a vector that has that balance where it can be highly infectious, but like Sean said, you actually remove all the bad stuff from it, but only leave the proteins that are important for being infectious. Right? So they can get into cells, but they really can't replicate any of the rest of the virus because it's been removed. The only thing it can replicate is the stuff you put in.
Okay.
Could you then make COVID a vector?
I bet you somebody's working on it. I bet you money somebody's working on it. They have to, because it is good. The problem is that those type of viruses are not good at getting into neurons. The best vectors we found for getting into neurons are lentiviruses, which are coronavirus is a retrovirus. It's RNA based. It's not DNA based, which is why it mutates so fast. But it's not as good at getting into neurons as something like HIV is, which is a lentivirus, a type of retrovirus. Yeah. And neurons, a lot of the diseases that we care about have neurologic effects that are the worst part of them, right? Like if you have Parkinson's disease, it's annoying to have the chorea, the movement like that, but it's the thing that kills you is actually the neurological degeneration. So you need a vector that can actually get into brain cells. Cool? All right. Okay, well, if you have to go meet with your other teachers, do that. I'm going to make, because office hours run for another half an hour, I'm going to make a list. I'm going to actually go into the calendar and put these on. And now that these are done, I can go and schedule who's going to do the tea times or other virtual things. And again, I'm also I want to see if we can go to the Museum of Natural History in New York City. I talked to Miss Bauer, usually runs a trip that I guess in the past has been a combination of the science department and theater for some reason that goes to the Smithsonian. So I think we want to do that too. And then any suggestions you have for other activities that you might want to do, we should try to plan it. Okay, let's we're in it now. So the second quarter starts. That's I think when I always want to start AMI. Let's really get it going. Freshmen, I hope. Listen, freshmen, you can ask any of the upperclassmen who are willing to help you because they're nice people. If you're confused, that's fine. You're supposed to be. That's how we all start. And what happens is over time you get used to this, and you get used to how to read a paper and how to talk about it and how to ask the right question and how to be confident. So you keep coming to this, you'll get better at it. And then, you may not appreciate it now, but when you get to college, you'll be way better suited to understand the real literature. He's waving? I think he's waving. Okay. No further comments or questions? Good job, Sean. Yay. All right. See you guys later. Happy Thanksgiving, everyone.
So happy Thanksgiving.
Bye-bye.
Happy Thanksgiving.
Thank you.
Happy Thanksgiving.